Hello, everyone, and welcome to today's keynote webinar titled Molecular Pharmacology Profiling, the Precursor to Preclinical Development. This webinar is a part of the 6th annual event in the Drug Discovery and Development Virtual Event Series. I'm Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. I now present today's speaker, Professor Kevin Pfleger, Director of Biomedical and Health Innovation at the University of Western Australia and WA Life Sciences Innovation Hub. He's also President of Australasian Society of Clinical and Experimental Pharmacologists and Toxicologists, Head of Molecular Endocrinology and Pharmacology at Harry Perkins Institute of Medical Research, as well as Deputy Director and UWA Node Leader at ARC Center for Personalised Therapeutics Technologies. For a complete biography on our speaker, please visit the Presenter tab from the menu at the left of your screen. Professor Pfleger, you may now begin your presentation.
Thank you very much, and it's an absolute pleasure to be here. If you have a validated drug target, you can run a high-throughput screen to find some compounds, or you can look for repurposing some compounds that are already known to hit that target. But once you have those compounds, and you may have a number of hits from your primary screen, it's a very good idea to actually profile, to actually see what pharmacology exists in the cell. What does activation of that receptor do? As well as ligand binding, you can look at activation of various different signaling pathways. Do you see ligand bias? Do you see activation or even inhibition of different pathways? Is there an effect on trafficking, both internalization and forward trafficking? There are so many things that you can look at to actually differentiate between different compounds, so that when you go into a preclinical development, you can actually differentiate between these compounds. It's also very important to consider kinetics. A lot of pharmacology is about what happens over time. So instead of taking snapshots, it's very important to do real-time assays so that you can actually understand what is happening over longer timescales. So what do I mean by molecular pharmacology profiling? Well, there's a number of different assay technologies that can be utilized, and it's about looking at the different facets of the signaling and also the binding. So looking at receptor ligand binding assays, using various different assays to monitor different signaling pathways. If you're looking at G protein coupled receptors, for example, that could be looking at G proteins. Receptor tyrosine kinases could be interactions with such molecules as GRAB2. And it's also looking at trafficking, because as a receptor is activated, it will usually translocate into the cell, internalize, and also the cell will respond to that activation by forward trafficking other receptors up to the cell membrane to enable that response to continue. So understanding the different profiles that these compounds have can actually give you some great insights. Often these receptors don't work in isolation. In fact, very rarely. They work in large multi-molecular complexes. And technologies such as Receptor-HIT, the Receptor-Heteromer investigation technology from Dimerix, can actually look at these complexes, investigate heteromerization, and understand how we can have heteromer-specific pharmacology. And then if we're looking at endogenous proteins, we can utilize CRISPR-Cas9 and actually study those endogenous proteins under endogenous regulation. And we can use that to monitor protein-protein interactions, receptor trafficking, and ligand binding. Bioluminescence resonance transfer, or BRET, is a technology that's now widely used across the field. And it's a way of looking at protein-protein proximity and also ligand receptor proximity. It enables us to actually, in real time, look at how these signaling pathways are activated. So we have a luciferase, we have a molecule that gives off blue light when it metabolizes its substrate. That is attached to a receptor of interest. We then have an interacting protein which is conjugated to a fluorophore, and that fluorophore acts as the acceptor for the energy transfer. When the receptor is activated by the ligand, the interacting protein comes up, interacts with that receptor, bringing the fluorophore into closer proximity to the luciferase. As a consequence, there's energy transfer. You have less blue light emitted and more yellow light or whatever color the fluorophore is. And as a consequence, we can then say that we have increased protein-protein proximity, or indeed ligand receptor proximity if we're looking at a binding assay. A BRET assay involves transfecting cells. This can be done in a microplate reader and often at 96 wells, but could be used at 384. And the microplate reader has filters so that you can actually look at the different colored light windows to actually understand how much light you're seeing emitted from the donor versus the amount of light emitted by the acceptor. So BRET receptor ligand binding assays. This was some work published and worked up in collaboration with Pharmacology and also Stephen Hill's laboratory in Nottingham in the UK. And you have the Nluc luciferase attached to the N-terminus of a receptor, or a GPCR in particular, and that's on the extracellular surface. And BRET occurs between that luciferase and the fluorescent ligand. So you have the acceptor conjugated to a ligand, and that is the energy transfer occurs to that BODIPI, for example. You can then compete that off with unlabeled ligand. Over here, we can actually see on the right-hand side of the panel here, we have two different acceptor fluorophores. So we have one that's more green, which is the BYFL, and one that's more red, which is the BY630. Now you can see you get quite similar pharmacology with the concentration response curves. The difference is the background on the raw BRET ratio. So you can see there's much more background with the BYFL because it is more green, and you can see the axis actually starts at 0.1, whereas with the BY630, which is more redshifted, there's much lower background. So we tend to use more redshifted fluorophores in these binding assays because of that much lower background. These binding assays can be used in saturation mode and also in kinetic mode. And the great thing about using a luminescence technology is that you have many orders of magnitude, which you just simply couldn't achieve with radioactivity. It would be too dangerous. You can also do kinetic work. So this is looking at the binding across 60 minutes, which gives you similar KDs to the saturation assays. This work can also be done with peptides as well as small molecules. So here we have the angiotensin 2 conjugated to TAMRA and giving us some nice pharmacology. Now looking at receptor signaling and regulation assays. Here we have arrestin, and those of you that work on G-protein coupled receptors know that arrestin is a very important molecule. Here we have a receptor the vasopressin receptor 2, which has a number of point mutations. And you can see this changes the kinetic profile. Again, this illustrates that the BRET technology, looking in real time, you can actually assess these different profiles, and this gives you important information. Now, these are point mutations of a receptor, but you could expect to see similar bias in terms of these profiles if you're looking at a range of different compounds. And some of those compounds may give you different profiles. Looking at arrestin assays here with the orexin receptors 1 and 2, this illustrates that you can actually look at kinetics. And if you were to just look at these experiments after about a quarter of an hour, they would look very similar. However, if you run the experiment for 240 minutes here, you can actually see that there's a distinct difference in the profiles. And we followed up these experiments with another study where we determined the reason for this prolonged profile was due to some differences in motifs in the C-terminal tail. So it's important to be able to look at these profiles, differentiate different pharmacology, and again, this can be seen with different compounds potentially. Here we illustrate looking at the localization of the G-alpha I unit and the G-gamma 2 unit, so 2 units of the heteromeric G protein complex. And you can see here that gauging the proximity of these two subunits gives you a readout of activation of the CCR4 chemokine receptor when activated by CCL22. We can also look at ubiquitin. So this is looking at the arginine vasopressin induced ubiquitin recruitment to the vasopressin receptor 2, both the wild type and mutants, and you can see some different profiles there. And then also on the right-hand side, looking at the orexin-induced interaction between arrestin and ubiquitin. So these are orexin receptors, and you can actually see if you tag the arrestin molecule with Nluc and ubiquitin molecule with venous acceptor, you can see a change in the proximity of those two when you activate the orexin receptors. And that is distinct if you have point mutations of those receptors. So moving now to the BRET trafficking assay. And this was work developed by Neven Lambert and his laboratory to actually monitor the internalization of receptors into the cell, utilizing various different markers. So we have KRAS as a fragment that is in the plasma membrane. And as the receptors move off the plasma membrane, the proximity to the KRAS reduces. You then have various different RABs, which are in the different compartments. So RAB5 in early endosome, RAB4 with rapid recycling, RAB11 going through the recycling endosome, RAB7 in the late endosome and lysosome, RAB9 then trafficking across to the Golgi network. Now we also add on to this some other RABs, which particularly looked at the forward trafficking, and indeed the RAB9, but also RAB8, RAB6, and RAB1. So this is looking at the forward trafficking from the ER up through the Golgi complex and up to the plasma membrane. And in combination, we see some very interesting changes depending on the compound that is used. This was all validated using confocal microscopy to demonstrate proximity to established markers of these different compartments. This slide shows the vasopressin receptor 2 trafficking with various different mutations, and it just illustrates the kinetic profiles that can be established over time, in real time. Remembering these are cell populations, but you can actually see quite distinct profiles with the kinetics changing depending on when the proximity changes. So we have RAB5 in early endosome happening rapidly, whereas the RAB11, for example, is slower because that is going through a slow recycling compartment. Moving now to the BRET Receptor-HIT assay, so the Receptor-Heteromer investigation technology. And just a little bit of background, a group came together Bethesda at NIH in the end of 2008 to discuss and establish the nomenclature for the field of receptor heteromers. And that group came up with this definition that a receptor heteromer is a macromolecular complex composed of at least two functional receptor units with biochemical properties that are demonstrably different from those of its individual components. So that means that a receptor heteromer is a large complex, often has many proteins involved, structural proteins, scaffolding proteins, as well as signaling proteins. And to be defined as a receptor heteromer, just two of those receptor units need to be functional receptor units. Now, the allosteric modulation between those receptor units, which enable it to have distinct pharmacology from its individual components, can occur either directly or via other receptors, if there's more than two receptors in the complex, or indeed through adapter proteins, which can connect the two receptors. So the two receptors themselves don't actually physically touch, but the allosterism is transferred. So what are the implications of GPCR heteromer activation? Well, if we run screens of a GPCR in isolation, we'll see an established effect for that GPCR activation, no doubt. However, if these receptors are in complexes, which indeed they are in the cells in our bodies, the pharmacology can change. And we have evidence now across the literature that transactivation can occur. Other receptors in the complex can be activated instead or in addition. And indeed, we can even have different effects, which I've labeled here as effect AB, which is novel to that particular complex. So we've changed over time. We've changed our thinking. We've gone from talking about a GPCR having a particular G protein mediated pathway, so talking about angiotensin receptors being GQ coupled, for example, to this idea that each individual receptor can actually potentially couple through different G protein mediated pathways at the plasma membrane, but can also potentially activate arrestin mediated pathways inside the cell. And whether those arrestin mediated pathways also involve G protein or not is up for debate. However, there does seem to be this potential for different activation of different pathways and indeed potentially inhibition of different pathways as well. So this is, of course, is the concept of biosignaling. But just to overlay that, if we have different complexes involving various different GPCRs and other molecules, those complexes can have different pharmacology. So there could be different coupling to G proteins at the plasma membrane. There could be different coupling to arrestin and those arrestin-mediated pathways as a result, which again may or may not involve G proteins inside the cell. So the consequence of this is that we also see biosignaling through heteromerization. And that means that we can potentially look for pharmacology where we have reduced side effects. It could be that a side effect of a drug is actually as a consequence of this transactivation through the complex. We can also look to potentially increase selectivity, because it may be that in certain tissues there may be a complex and in other tissues there isn't. So if we can understand the heteromerization and the impact on pharmacology, we can potentially target tissues where there is a particular heteromer, or indeed where there's an absence of a heteromer, to actually target our therapeutic more effectively. The issue, though, is that understanding the pharmacology of heteromers is technically demanding. What we really need as a field is to develop tool compounds using these sorts of systems in model systems, to then take those tool compounds into in vivo settings to understand the physiological relevance of these heteromers and really get to grips with the role they play in pharmacology. The technology, the receptor-heteromer investigation technology that I mentioned before, is an approach and a configuration to help us investigate these complexes. So we have a GPCRA, or it could be any receptor, but I illustrate here with G protein coupled receptors. That first receptor is tagged to a first reporter component. And that reporter system could be BRET or FRET or enzyme fragment complementation or split luciferase or split fluorophore or Tango, the Dimerix system. There's many different systems that look at proximity. So we have the first part of that reporter component attached to that first receptor. We then have GPCRB, or the second receptor, unlabeled with respect to that reporter system. We then have our second reporter component attached to the interacting group, and that interacting group translocates up and interacts with the complex when you activate that GPCRB or the heteromer complex. So that means that the second reporter component is brought into closer proximity with the first reporter component, and as a consequence, we have a signal. This can be done with an interacting group that's inside the cell, such as beta-arrestin or G protein or GRAB2. It can also be configured the other way up, where we have the first reporter component labeled on the N-terminus of GPCRA, and the second reporter component is attached to a ligand. So this means that you have a fluorescently labeled ligand with the fluorophore being the second reporter component. You can then look at a range of different compounds that are unlabeled. They would then compete for the interacting group, and this would actually come away from the receptor. The proximity between the first and second reporter components would reduce, and as a consequence, we have a competition binding assay that is selective for the heteromer complex. This is illustrating receptor-HIT configuration, but this time particularly with the BRET. And here we have the Nluc luciferase labeled to receptor A or connected to receptor A. We have receptor B that's unlabeled, and then we have the YFP, yellow fluorescent protein, attached to the beta-arrestin molecule. When we have a ligand that's selective for receptor B or the heteromer, there's three possibilities. The receptor A Nluc homers are not going to give you a signal because there's no ligand binding and therefore no recruitment of the acceptor. Receptor B homers similarly won't give you a signal because they don't have the donor present. So even though you get recruitment to the acceptor, there's no donor to result in the signal. So it is the heteromer complex that gives you the signal, and this leaves the other two in the dark and gives you that clean signal that is due to the heteromerization. Just to illustrate some of these data, and this is data that underpins the DMX200 that is now being translated by Dimerix and is in phase 3 clinical trials for chronic kidney disease, focal segmental glomerulosclerosis, and also COVID-19. So here we have the CCR2 receptor. We have a change in the proximity of the donor and acceptor when you have activation of this receptor, which changes the proximity to the G alpha I. And you can see here you have the concentration response curve when you activate with various concentrations of CCL2. Now, if you add angiotensin 2 of various different concentrations to a maximum dose of CCL2, in the absence of the angiotensin receptor, then you don't see any change in that across the concentrations, and this is in panel C, of course. Now, in panel D, in the presence of the AT1 receptor, we still see the concentration response curve changing when we have CCL2 activating CCR2. However, interestingly, when we have a maximal dose of CCL2 and then we have a range of concentrations of angiotensin 2, in the presence of the AT1 receptor, we're now seeing a reduction in that change in BRET signal. So we're seeing a reversal of that activation of CCR2. So this is interesting because a number of instances where we have high blood pressure, for example, angiotensin receptor blockade is a frontline treatment. Obviously, in a number of inflammatory disorders, also angiotensin blockade is utilized. However, in that situation, it's also useful, according to these data, to actually block the chemokine response as well, because even though you're looking to reduce inflammation by blocking the angiotensin receptor system, you could actually be unmasking a chemokine response in doing so. So if you can also block the chemokine pathway, then that will be a better outcome in terms of anti-inflammatory outcome. Here we can see there's also recruitment of beta-arrestin to the CCR2, and in the presence of the AT1 receptor activated by angiotensin 2, we see this receptor-HIT signal. And interestingly, if you activate with both angiotensin 2 and the chemokine CCL2, you get this more than additive response. And this is indicative of heteromerization. These slides here illustrate that if you block signaling of one of the receptors, we reduce the signal, and it's when you block both receptors in combination that you reduce the signal right down and inhibit that beta-arrestin recruitment. So again, hinting at this dual receptor blockade being of benefit. Now, the receptor for advanced glycation end products is not a GPCR, but it's there's evidence that we've found that it actually is transactivated by a range of GPCRs. So here we have some data with the angiotensin receptor AT1. We have the receptor-HIT signal. So as controls, we can illustrate here that the angiotensin receptor gives a strong recruitment of beta-arrestin 2. The CCR4 receptor also gives a strong recruitment of beta-arrestin 2 when activated by CCL22. However, when we look at the RAGE Biotech recruiting arrestin proximal to it, that occurs when you activate the angiotensin receptor with Ang2, recruiting the arrestin proximal to RAGE R-Leucate. However, the CCR4, even though it recruits arrestin well, we do not see proximity to RAGE when you have CCL22 activating CCR4 in the presence of RAGE R-Leucate. So that's a nice negative control, and illustrating that not every receptor seems to have this effect with RAGE, but certainly the angiotensin receptor 2 does. Now, panel E, you can see some various different proximity markers that enable us to monitor trafficking. This is an interesting panel because we usually see when we activate a receptor that the signal with KRAS goes down because the receptor is internalized and the proximity between the KRAS marker and the receptor goes down. However, interestingly, for this RAGE interaction, the KRAS proximity goes up. Now, you could be forgiven for wondering why that would be, particularly as you can see here that the RAB5 signal goes up, so the KRAS is translocating into the RAB5 compartment, which is the early endosome. It's also going through the RAB11 compartment, which is the recycling endosome. So we are seeing internalization from the membrane, but interestingly, we are also seeing forward trafficking because you can see the RAB8 proximity increasing. So we have this process where not only are we having the RAGE internalize, but we're also seeing it recycle up back to the membrane, and also we have increased forward trafficking. So the end result of that is an increased presence of that receptor on the plasma membrane, proximal to the KRAS fragment. Not only can we use G-protein-coupled receptors, but we can also study receptor tyrosine kinases and other receptors. And this is utilizing GRAB2 Venus as the intracellular interacting protein. So similar idea, here we would have a ligand selective for the unlabeled receptor tyrosine kinase 2, and as a consequence, we don't see any BRET with our TK1 homers because we don't have recruitment of the acceptor. We don't see BRET with the RTK2 homer because we don't have a donor present, and the signal comes from the heteromer complex, where we're complexing with the GRAB2, bringing that acceptor to the complex. Here's the pharmacology we would see. So we see EGF activating EGFR. We see heregulin also activating and recruiting this GRAB2, but only actually in the presence of EGFR, as you can see in this panel D. This is an interesting complex and some interesting pharmacology that's well established with this family. But certainly in panel C, you can see that if you have EGF activation of EGFR, that occurs across and gives you this receptor hit signal. This is the VEGFR2 heteromerization with the B2 receptor, and this is work from Steve Hill's lab that we collaborated on. And this is interesting because it shows you the power of kinetic profiles and why it's really important to do kinetic profiles. Again, if you were to just run these assays and look at the endpoints at about 10 minutes, they would all look very similar. Here we have isoprenaline activating the B2 adrenergic receptor, recruiting arrestin to that receptor, bringing it into close proximity with the Nluc there. Now we have that's labeled to the B2 adrenergic receptor. Now we see that effect. So if you look at panel B, we see the isoprenaline giving us this nice response with the blue circles. But interestingly, if you also activate the VEGF receptor 2 with the VEGF165A, you get a change in the profile. So the red line, you can see a very different kinetic profile when you have the combination of isoprenaline and VEGF165A. And that profile is reversed if you then antagonize the VEGF165A with sedaranib, restoring that profile back to what you see with the isoprenaline on its own. So you can see some really nice pharmacology if you run these experiments over time and establish the kinetics. This is the receptor-HIT ligand binding assay. So here we have Nluc attached to the B2 adrenergic receptor on the N-terminus. We have a BRET signal going from that Nluc to a Biodipi labeled propranolol, and that's competed off with unlabeled propranolol. And you can see a nice saturation curve there. Now, if you have the Nluc attached to an angiotensin receptor, and you now have the B2 adrenergic receptor unlabeled with respect to the donor, when you bind the Biodipi propranolol to that B2 adrenergic receptor and compete it off, we're now seeing this saturation assay with the BRET going across the complex from the Nluc to the Biodipi propranolol on that adjacent receptor. This gives you a nice saturation curve, but notice that it is about 1/10 of the strength of the curve at the top of the graphs, top of the panel here. And that's because when you have energy transfer, it is highly distance dependent. So it's inversely distance dependent to the 6th power. So that energy transfer across the complex it results in a much lower signal. And this last part of the talk, what I'd like to do is just take you through some of our work with the CRISPR-Cas9. And this is particularly work from Carl White, who's a postdoc with me and now works with Dimerix. The aim here was to actually look at endogenous chemokine receptors. So this is the endogenous CXCR4 that's in HEC293 cells, and actually using CRISPR-Cas9 to engineer the Nluc onto that endogenous CXCR4, which means that it's still under endogenous promotion. We then transfect in exogenous beta-arrestin to venous and look at the outcomes in terms of the BRET signal. Interestingly, we still see a nice signal. So we have the CXCL12 activating the CXCR4. We see quite a transient response, and that may be more transient than we sometimes see with, or we see with overexpression systems. So again, illustrating that it's good to use these low expression levels if we can. And this signal is reduced and inhibited by the antagonist AMD 3100. You can see some nice pharmacology here with a PEC50 of 8.6. Z prime data. So this is for those of you that aren't familiar, this is a measure of assay performance. And we have very strong Z prime. So an ideal Z prime is 1, but anything above 0.6 is excellent. And we have some very good Z factors for the beta-arrestin 2 labeled with Nluc. So this is endogenously expressed under endogenous promotion. And then the transfected vasopressin receptor 2 that's labeled with venous. So if you've got 25,000 cells per well, we have a very good Z factor, and this is even better if you have 50,000 cells per well. Now, if you compare that to the beta-arrestin 2 the beta-arrestin 2 Rluc 8, which has a much lower light output, the Z factors are negative, which is meaningless. So, and you can see the overlap of the different points. So this illustrates that if you are wanting to do assays with very low expression levels, it's a good idea to use an Nluc because of its brightness. In terms of trafficking, here we have the KRAS labeled with venous. So this is the plasma membrane marker. We have the RAB5 with the label with venous label, and the Rluc, the Nluc again on the endogenous CXCR4. And you can see we have this reduction in the proximity to KRAS as the receptor goes into the cell, and we have the increase in proximity to the RAB5 in the early endosome, again as the receptor goes into the cell. We then have proximity to RAB9 labeled with venous as it goes into the late endosome, RAB11 venous in the black open circles here underneath the red, with the RAB11, and then we have with the blue upside down triangles, we have the RAB7 venous as we get translocation to the lysosome. So just illustrating that you can perform this trafficking assay with the endogenous protein tagged with the BRET donor Nluc. And then you can also multiplex. So here we have the Nluc labeled CXCR4 endogenously, and then we have the KRAS labeled with halo tag, which is red, and then we have the RAB5, which is labeled with venous. And using a microplate reader that can actually measure multiple channels, so the Claristar, we actually see this reduction in the proximity to the KRAS, and with the same cells, we can see the increase in proximity to the RAB4 venous. We can perform receptor-HIT with this endogenous CXCR4 tagged with Nluc using CRISPR. Here we have the transfected in GPCR with transfected in beta-arrestin 2 labeled with venous, but the CXCR4 is endogenous labeled with Nluc with CRISPR. We can see in the graph here that if you activate with CXCL12, we see this transient response similar to what we were seeing with previously. If we then activate with isoprenaline, you can actually see this signal in the green, and you're actually seeing the recruitment of the arrestin to the beta-2 adrenergic receptor, bring it in a close proximity to the CXCR4 labeled with Nluc. Interestingly, when you add both ligands, you get this more than additive effect. So again, implying that we have heteromerization, and this is a heteromer that's been published previously. The Nluc ligand binding assay. Here we have the actual illustration here in panel A, where we've actually put that Nluc in front of the A2B receptor. If you look at panel B, the graph on panel B, we have very, very low luminescence when we have the Nluc labeled A2B. When that's introduced with CRISPR, that's a similar level to using 0.05 nanograms in a transient transfection. That's incredibly small. You compare that to the 5 nanograms, which is also pretty small, but 5 nanograms gives you a much, much stronger luminescent signal. The levels we are talking about when we're using CRISPR are very low in comparison to the normal overexpression systems that we use in the laboratory. However, we get similar cytokine P accumulation, and so we get the similar profiles there. And we get very nice saturation data. So you can see here with panel B, we have a very nice saturation assay competed off with the selective compound. If you have a non-selective or a compound that is not selective for that particular receptor, we don't see that effect. However, as you increase the concentration, you start to see some inhibition there. With panel E, we see that there's no effect with the TAMRA Ang2, which is obviously not the correct ligand for this receptor. And we can also see this with both agonists and antagonists. We can illustrate this with kinetics. So again, we can establish KDs using this kinetic profile, and this generates some really nice concentration response data, as you can see in panel B. Remembering this is with very, very low expression of receptor in these CRISPR cell lines. And if you compare that to what we would see in transient transfection, at the top panel here we have 0.05 nanograms. So that gives you, still gives you a nice saturation, but the kinetics, you can actually see it's a little bit messy. And if you look at the concentration response curves, again, a little bit messy. Much, much cleaner, of course, with the 5 nanograms with really nice data. But I'd like to just sort of draw attention to the comparing the 0.05 nanograms, where you've got some quite messy data there, with the previous slide, where you have similar expression levels, but because it's CRISPR and you have receptor in multiple cells, then you have very nice clean concentration response curves. So illustrating again that the CRISPR is the way forward if you want to look at low expression levels. So I'd like to finish up now with some acknowledgements. This work was carried out in the Molecular Endocrinology and Pharmacology Laboratory at the Harry Perkins Institute of Medical Research. My colleagues have done the work, so Liz Johnson, Carl White, Ruth Sieber, Ethan C., Repti Apawadana, and Tasha Dale. Some selected past colleagues as well that I'd like to call out. So I've shown their work here with Mohamed Akli-Ayub, Matthew Dalrymple, and Werner Jaeger. A number of academic collaborations from all over the world, and indeed industry collaborations as well, particularly with Promega with BMG Lab Tech instrumentation, with of course Dimerix and more recently Accelerate. And I'd like to acknowledge the funding from the Australian Research Council, the National Health and Medical Research Council, Dimerix, Brain Foundation, and then our partners for linkage grants, Promega, BMG Lab Tech, and University of Nottingham, and more recently the Diabetes Research WA. Thank you very much indeed. I'll leave you with my contact details. Thank you.
Thank you, Professor Pfleger, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until February 22nd, 2025. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.